All right, so last thing we're going to do is talk about a few polysaccharides. We're really going to just learn some fun facts about the most common polysaccharides. First of all, polysaccharides are, as we've talked about before, they're now connecting more than two monosaccharides together. They're used for both storage of sugar to be used as energy, as well as structural features. So it turns out that cellulose, which is a main component of wood, as well as cotton, these very structurally sound materials, the main structural element of there is actually a polysaccharide. Okay. Polysaccharides are joined when monosaccharides are linked together, and there are many of them. Okay, so we're going to talk about each one of these: amylose, amylopectin, cellulose, and glycogen. We're going to learn a little bit about each of them. The good news is they are all polymers of D-glucose. So the same sugar is being used in all of them. Okay, starch is what we call a polysaccharide that's made in a plant that's used for energy. Okay, so plants store their sugars in starches so that later on they can break down those starches and use those sugars for energy. And there are two main types. The first one is amylose. Okay, amylose is an unbranched chain. Okay, so it's a chain of glucose linked together all in one line. And specifically, they're linked by alpha-1,4 glycosidic bonds. So hopefully everybody can be convinced that this is an alpha glycosidic bond, and it takes place between carbons 1 and carbons 4 of neighboring glucose. So it's just a chain of a bunch of glucose molecules bound together by alpha-1,4 glycosidic bonds. All right, and again, this is it's a starch, meaning that it's made by a plant, and it's used for energy. It turns out that it's actually not the most common starch in plants. It makes up about 20% of starch within a plant, is this unbranched amylose polysaccharide. The other 80% is made up by amylopectin, which is very, very similar. It's connected by alpha-1,4 bonds, but it's not a straight chain, it's branched. And those branches are made of alpha-1,6 glycosidic bonds. And they're about every 20 to 25 units of glucose. Okay. They're easily hydrolyzed into individual sugars. They actually first go to maltose, which we talked about were those disaccharides, and then finally to glucose. But so here's an example of amylopectin that has both of these alpha-1,4 glycosidic bonds, so it's like these, but then it's got these branch points every few carbons. That's an alpha-1,6 glycosidic bond. So hopefully you can convince yourself that this is indeed an alpha-1,6. Whoops, let's just go through, make sure we got that. Here's my anomeric carbon. That oxygen is pointed downwards, making it an alpha, and then it's connected to carbon 6 on the neighboring carbon, right? So an alpha-1,6 glycosidic bond. So you have these straight chain amylose fibers, and then if there's branch points every so often, it becomes an amylopectin, and the straight chain is all alpha-1,4 glycosidic bonds. Those branch points are alpha-1,6 glycosidic bonds. Okay, and that's how plants store their energy, right? So they make all this sugar based on the sunlight, and then they store that sugar for later, and they do so about 20% in amylose and 80% in this amylopectin. So if you take a really starchy vegetable like a potato, that starch in there are all amylose and amylopectins. Okay, and again, they're used for energy. So that's the purpose of amylose and amylopectin, is the plant will later break it down into components when it needs energy, right? And so both of these can be broken down first to maltose, which is just that glucose disaccharide, and then into a ton of different glucose units so that it could use that for energy, right? Further digest those. In animals, we don't have starches. We have glycogen, okay? Functionally, it's basically the same thing, but when it's an animal, we don't call it a starch. So it's often called an animal starch, but it's really glycogen, okay? Because we just don't call them starches when they're in animals, okay? It's very similar to amylopectin. It consists of the alpha-1,4 linkages and then these alpha-1,6 branch points, okay? The only difference is it's slightly more highly branched than amylopectin. Whereas in amylopectin, a branch point would occur every 20 to 25 glucose units. For glycogen, it occurs every 10 to 15 glucose units, every 10 to 15 glucose molecules. Functionally, though, I mean, it's you could think about it as an animal starch. That is kind of how it's thought of, right? It's another just branched polysaccharide, and it's how we store our energy. So, right, we talked about how your body likes to maintain a constant blood glucose level. Well, how it does that is when it has extra glucose, it'll make glycogen. And then if you haven't eaten in a while, it will start pulling from those glycogen reserves, and all in this delicate balance that keeps your blood glucose levels pretty much constant. The last one is also a polysaccharide that's found in plants, but it's not used for energy. It's used for structure. It's what's called a structural carbohydrate, and that is cellulose. And cellulose is made of unbranched chains with a beta-1,4 glycosidic bond, right? So very similar to amylose, it's just one long fiber of glucose units stacked together, but instead of being joined by alpha-1,4 glycosidic bonds like amylose, it's beta-1,4 glycosidic bonds. It forms this really rigid structure, and what's more is that human beings and most animals can't digest that beta-1,4 glycosidic linkage. We don't have an enzyme that's able to break it apart, which is why if you ate a piece of wood, you wouldn't be able to digest it into energy, right? We can't eat trees and gain energy from it. Trees mainly package all that stuff into cellulose, and although it consists of a bunch of glucose, our body can't figure out how to get the individual glucose molecules from that chain. Okay, so here's an example of this beta-1,4 glycosidic linkage. Let's again just kind of practice this to make sure we got it. Here's my anomeric carbon, and it is indeed in that beta form because that oxygen's pointed up. So this is a beta-1, and then we can count to make sure 1, 2, 3, 4. So this is indeed a beta-1,4 glycosidic linkage. Okay, here's like the cheat sheet of what we need to know about polysaccharides. Amylose, less abundant plant starch made up of glucose units and the alpha-1,4 glycosidic bond. Amylopectin, more abundant plant starch than amylose, also made up of glucose units, and it has those straight chain 1,4 alpha-1,4 glycosidic linkages, as well as these alpha-1,6 branch points. Glycogen is the animal starch, but we don't call it a starch, we call it a glycogen. And it's also made up of glucose units. It's a lot like amylopectin in that it consists of alpha-1,4 and alpha-1,6 glycosidic bonds, but it is slightly more branched than an amylopectin. And then lastly, cellulose, which is not a starch because it's not used for energy. It's a structural carbohydrate. It's still made up of glucose, but they contain these beta-1,4 glycosidic bonds that our body and most animals don't know how to digest.